Hi everyone, and thank you for listening to my presentation. Today I will be discussing my project that focuses on the role of a viral protein, ORF20, in Kaposi sarcoma-associated herpes virus during lytic infection. Kaposi sarcoma herpes virus, also known as KSHV, is an oncogenic virus, meaning it is capable of causing cancer. Specifically, KSHV infection is the causative agent of several malignancies, including Kaposi sarcoma, primary effusion lymphoma, and multicentric Castleman disease. Kaposi sarcoma, its namesake disease, is the most common and aggressive cancer in HIV-infected, untreated patients, and it's pictured to the left. The KSHV genome, pictured on the right, is a double-stranded DNA circular genome that's about 165 to 175 kilobases, and it consists of over 85 possible genes. The KSHV life cycle consists of two phases, so there's the latent and the lytic phases. The latent phase occurs when the virus is dormant in the cell, so it's trying to remain undiscovered by the host immune system. The lytic phase is a productive phase, so when viral genes begin to be replicated, virions are produced, and it begins infecting other cells. Widespread changes of the host nucleus and global nuclear reorganization occur upon lytic infection. And this reorganization includes the formation of these viral replication compartments, which are these subnuclear structures where the viral DNA genome can be replicated. Lytic replication proceeds distinctly with a rolling circle mechanism where the concatenameric genomic DNA is produced. My project in particular is focused on KSHV ORF20, which is a largely uncharacterized gene, but one that we believe plays a role in lytic infection and replication. Upon initiation of the lytic phase, transcription of viral genes begins following a cascade, with first being immediate early genes, followed by early genes, and lastly late genes. Immediate early genes are associated with the production of transcriptional and regulatory viral proteins. Early genes are responsible for viral DNA replication and protein production preparations, and late genes are subsequently associated with viral DNA replication and viral structural components. We first characterized the expression kinetics of ORF20 by comparing the relative levels of mRNA made in the cell at 4 different time points, so 0, 24, 48, and 72 hours after lytic infection was induced. And we compared these levels of ORF20 to other known genes via RT-qPCR, which is real-time quantitative polymerase chain reaction. So we looked at the expression of ORF20 compared to the expression of ORF37, a known early gene, and the expression of K8.1, a known late gene. And the expression of ORF20 is comparative to that of ORF37, the known early gene, suggesting that ORF20 itself is an early gene as well. From here, I looked at what other proteins ORF20 interacts with. So we wanted to characterize all the protein-protein interactions, including the transient ones, which is why we used a promiscuous biotin ligase that allows us to capture even fleeting interactions with ORF20 and see what proteins those are and capture it via mass spectrometry. So we found that ORF20 interacts with several host proteins that are involved in replication. And then additionally, we saw that ORF20 interacts with another viral protein, ORF59, which has previously been known to be a DNA processivity factor. From here, we decided to look at the subcellular localization of ORF20. So as discussed, KSHV lytic infection requires remodeling of the host nucleus and the formation of these replication compartments where the DNA is replicated. These are visualized in the cell as dark spots that we outlined with little white dots, and they are in the nucleus when stained blue with DAPI. So to examine where ORF20 localizes to in the cell, we first established a cell line that is deficient in ORF20, and we also used wild-type KSHV infected cells. A tagged ORF20 protein was transfected into both these cells, which allowed us to visualize the location of this protein in both these cell lines via the red staining. So the blue staining is DAPI, which just stains the nucleus, and the red staining is our protein of interest, ORF20. And we have both wild-type and ORF20 deficient cells. So ORF20 in red is shown to be localized within these replication compartments in both these cells. From here, we used the ORF20 deficient cell line, and we really looked into how it differed in both gene expression and DNA replication as compared with our wild-type KSHV infected cells. So on the left, we're looking at two genes, so ORF59 and K8.1. ORF59 is an early gene, and K8.1 is a late gene. And we're looking at the expression of these two genes in our wild-type KSHV infected cell line and our ORF20 deficient cell line. So ORF20 levels are not changed when ORF20 is knocked out. Whereas in our late gene, K8.1, the expression of that gene is hampered when ORF20 is knocked out. And these levels of mRNA were captured via RT-qPCR. So essentially, in the cells without ORF20, late gene expression is hampered. Then on the right, we're looking at overall DNA replication. And we're looking at the same thing, so the levels of DNA replication when there is the wild-type KSHV infected cell line and when we have our ORF20 deficient cell line. And as you can see, without ORF20, DNA replication is diminished. So just to conclude here, we know that ORF20 is an early lytic gene. It interacts with host replication proteins and the viral replication processivity factor, ORF59. ORF20 also localizes to the replication compartments, and ORF20 is important for both late gene expression and DNA replication. So I would like to say thank you to my lab, thank you to my wonderful PI and mentor, Dr. Mandy Muller, and to all the graduate students in my lab, Will, Jake, and Dan, who have helped me immensely with this project. Thank you so much, and thank you all for listening.